Welcome to Teacher Sura channel of Learning Biology. For today we are going to study about food test and we are going to test direct in the laboratory. Welcome. This is Biology Form 4. We are going to test different food nutrients. Before we proceed, you have to subscribe, like and comment if you have any concern. Thank you and welcome. Before we proceeding to test, you have to understand that in food test we have different food nutrients that we are required to test in the laboratory. Number one, we have carbohydrates, which is divided into reducing sugar, non-reducing sugar, and starch. Also, we have protein and lipid. But for today, we are going to see different, we can call it 2 food nutrients, which we are going to test in the laboratory. Okay, before we proceed, you have to read this question from the blackboard, and then we are going to test according to the question as it's provided. Thank you, welcome. Let us test it together. After preparing solution, now we are going to test reducing sugar. Put the sample to the test tube. Because we are testing reducing sugar, we are going to add the amount of Benedict solution. This is our sample. Now we are going to heat to test the amount of Benedict solution. After putting Benedict solution, we are going to heat in water bath. Then the color will change from blue, green, yellow, orange, and finally black red. If the color of Benedict solution will change, it means that no amount of reducing sugar. This second test tube, we are going to test non-reducing sugar. We are putting amount of sample solution to the test tube. Because we are testing non-reducing sugar, we are going to add the amount of dilute hydrochloric acid to the sample solution. Then add. We have already added amount of hydrochloric acid. Then we are going to add amount of sodium. After adding amount of hydrochloric acid, we are going to heat. Then we'll allow our solution to cool for a few times, like 2 minutes. Then we are proceeding with another procedure. After putting hydrochloric acid, we are heating our solution for a few minutes. Then we allow it to cool and add the amount of sodium hydroxide and Benedict solution. Now we are going to add the sodium hydroxide. After cool, add a few amount of sodium hydroxide. Then after, add shake. We are going to add the amount of Benedict solution. Then we shake and heat. Okay, we are going to heat for the second time in order to see the series of color changing when we are testing non-reducing sugar. The color starting changing from blue to green, then yellow color, from yellow to orange, and finally brick red. Finally, the color will change to brick red. Then non-reducing sugar present. because our experiment was guided us to use chemical reagents provided to carry out the experiment to identify the food nutrient present in solution. Now, our reagents, we have Benedict solution, dilute hydrochloric acid, and sodium hydroxide. Because we have these few reagents now, that's why we have already tested only two type of food nutrients, which is reducing sugar and non-reducing sugar. Then after we are going to prepare practical report according to the question. Listening Teacher Sura channel of Learning Biology. Today we was discussing on how to test different foods in the laboratory, especially reducing sugar and non-reducing sugar. Thank you for watching. Welcome. Wish you can subscribe and like and comment if you have any concern. Thank you very much. I wish you goodbye.